Okay, hopefully everyone can hear me. Give me just a second to share my screen and kind of get everything ready, and then I will get going. Okay, so first let me pull up the PowerPoint. Again, there's so many buttons on here, and let's see. Okay, we'll start from the top. I have somewhere. Okay, so what we're gonna do is just kind of go through the review. If you want to have the review pulled up with you, that's great. If not, I'll switch kind of back and forth from it, or I'll just kind of tell you what topic we're discussing at the moment. So the first part on the Chapter 3 review is waste products. So with waste products, we're referring to what are not the byproducts of aerobic metabolism, but waste products. Means that they're not going to be used at all. And lucky enough for aerobic metabolism, these waste products are actually innocuous, meaning they're harmless. So if anyone wants to unmute their microphone and tell me what the two waste products are in aerobic metabolism, please go ahead. I will wait for an answer. Anybody? Someone's got to say before I move on. What are the two waste products of aerobic metabolism?
Is water one of them?
Yes, water is one of them. So water is a byproduct at the very end of aerobic metabolism. And water is, again, it's harmless. Our body uses water, so that water helps to regenerate the cycle over and over again. There's one more waste product. Does anybody know what that waste product is?
Is it carbon dioxide?
It is carbon dioxide, yes. So those are our two waste products. Where does the carbon dioxide come from? We'll look when we go over the energy systems again that carbon kind of splits off some of the reactions that occur. And when carbon splits off, it's going to combine with oxygen, forming CO2, which we just exhale. So that's kind of one of the first bullet points on that review is looking at the waste products. Next is structure of the mitochondrion. So on your assignment, you just wrote about the structure and the function or purpose of mitochondria when it comes to ATP production. So kind of what I'm concerned with for this is we already know it's the powerhouse of the cell. We also already know that all of these energy systems take place in the mitochondrion. But just kind of, we actually get a little bit more specific about what is the porin and what is the cristae and what is the matrix. And so it says that kind of on the review. And again, it's very, I know I said we're getting kind of more in depth, but it's not, it's basically just what I talked about in the lecture. So porins, we know, are these little, they're kind of passageways on the sides of the mitochondrion that allows for substances to be passed back and forth from the outside of the mitochondria to the inside. The cristae, we know, is this folded structure on the inside. This is it's a tightly regulated system that only allows for the passage of ATP and oxygen to pass through the cristae. And then the matrix, we know, is just kind of the space that is inside the cristae. And this is where the ribosomes are and the genetic material and the DNA. So yeah, that's really all you have to know, is just those three parts of the mitochondrion. Next, we kind of jump into the intermediate step. So we know that this step is integral to start the Krebs cycle, and we can't start the Krebs cycle without this intermediate step, because we know that pyruvate cannot enter into the Krebs cycle. So this is where we turn pyruvate into acetyl-CoA. So this picture here kind of talks about this conversion, it explains it, but this next slide does the same thing, and this one may be a little bit better to understand. We know that we have two pyruvates that were left over at the end of glycolysis. So this talks about one at a time, we're going to convert them into acetyl-CoA. So this process here, or kind of what the question is going to ask, is kind of what are the byproducts of this? How does this process work? So we know pyruvate here. During this process, we lose a carbon, which combines with oxygen, and we see that here. So we have CO2 as a waste product. And then we also had a hydrogen and its associated electrons split off of this reaction, or during this oxidation process, and it combined with NAD. So we have one NADH, and then the coenzyme A was added, making acetyl-CoA. Basically, the carbons just transferred over, and we added the coenzyme A, so having a 2-carbon molecule plus the acetyl group gives us acetyl-CoA. So what you basically have to know is there are 2 NADHs that result from the pyruvate to the acetyl-CoA, which is beneficial and will carry over to the electron transport chain, where we can actually use it to make more ATP. So that's basically this process here, and we already know that. This picture shows it. That intermediate step, we add a CoA, we lose a carbon, and we have one NADH. But since we had 2 pyruvates, we now have 2 NADHs. So that's really all that question, or that topic once, or just be sure you know that topic. Next, let's get into the Krebs cycle. So if we go down here, this here already kind of shows all of the steps that I talked about. And if you refer back to the lecture, I did go over these steps in a lot of detail. I talked about every single arrow and what happens when we transform one molecule and oxidize it into another. I'm not going to do that here, but if you want that review, just go back and listen to the video. But here, we're just mainly making sure that we understand what are the main byproducts that are beneficial for energy production in the electron transport chain. And we know that we have 2 acetyl-CoAs that are going to drop into this cycle. And again, it's called a cycle because we start and end with the same molecule, which is oxaloacetate. So oxaloacetate starts this cycle, and it will become oxidized and go through all these different reactions, and it will turn back into itself. So during this process here, we know that we have 2 CO2s produced, but we actually don't have to count those because, again, they're just waste products. We just exhale that. But let's talk about what we can use. So we see here we have 3 NADHs that were produced. They were produced when the hydrogen split off of the reaction and it combined with the NAD molecule. So those are going to transfer over to the electron transport chain. Same with this FADH. This FADH, we had 2 of them produced. Those are not as, they don't have as much energy as the NADH, so we don't produce as much ATP with those, but we can still produce a little bit, so still beneficial. And then we talked about this molecule down here, GTP, which is guanosine triphosphate. We said it's energetically equivalent to an ATP, but its uses are more restricted, meaning that it's mainly only used in muscle protein synthesis, or MPS, where ATP is used in a lot of other tissue in the body besides muscle. So GTP equivalent to ATP. So that's where our ATP was produced. Again, we had 2 cycles, so we have 2 ATP. So that's really what you have to know for the Krebs cycle is you just have to know those byproducts. You do have to know guanosine triphosphate and that it is the same as ATP, it's equivalent to it. So that word, if that comes up, just remember GTP, ATP, they are similar. Also, this slide here is important to know. There's going to be a question on there that's talking about all these byproducts before we enter into the electron transport chain. So you do have to be familiar with, if I start with glucose, how many ATP and NADH are produced in glycolysis, how many are produced in the intermediate, and how many are produced in the Krebs. You also have to be able to do that simple math and get our total so far after about 3 minutes of exercise before we enter into.
Well, I feel like last summer they had y'all conditioning like every single day. You know what I mean? Like it's not fair to just, I mean, they'd be like, okay, time to run your sprints.
Whoever has their mic not muted, just be sure
Yeah,
to
she's not taking any
keep
notes
your
on
mic
stories.
muted, but I'm going to keep going.
down.
Okay, so thus far, I know it says up here after 3 minutes of exercise, this is because glycolysis.
We could do it 4 times.
We've started glycolysis, and that was about 2 to 3 minutes
Oh, it's okay.
of energy production. And then before we enter into the aerobic system, we know we have to lower our exercise intensity in order to increase our exercise duration. And so that's what we're doing in
the second one.
TC. This slide here, just be familiar with what all these byproducts are and how much of each one of them have before the ETC actually starts working.
You rub it.
Okay, I'll say it one more time. If you haven't muted your microphone, please do so, just so everyone can hear me clearly. All right, so we're going to keep going, and now we're going to talk about the electron transport chain.
You see my bed all dirty?
So with our electron transport chain, we know we're in the mitochondria still. We know that the Krebs cycle was also in the mitochondria. And how you want to think about this is the inner and the outer membrane. So if you look out here, we have the outer membrane and we have the inner membrane. So we're really just talking about how we are pumping hydrogen into the outer membrane.
Energy production and electron
Hydrogen on
transport
the
chain.
inner membrane. And
What test do you think I
producing
made a 96 on yesterday?
Huh?
What test do you think I made a 96 on yesterday?
My history quiz?
hold on just a second, guys.
Yeah, you're real smooth saying, remind me.
Yeah, I know. I need you to remind me.
Okay, I hate to say this again, but
I am.
please mute your
Doesn't help.
microphones so I can continue to talk. Okay, so electron transport chain. I actually like this picture the best. It explains it. I know I've got 2 pictures on there. I feel like this one gives the most information and kind of helps you figure out what this purpose or what the purpose of this system is. But we know down here at the bottom, we're kind of in this inner compartment and we're pumping the hydrogens to the outer compartment. So first you have to remember, where did these hydrogens come from? NADH and FADH. So NADH can drop their hydrogens off at this first pump here. And when they drop their hydrogens at the first pump, those hydrogens are pumped now to this outer membrane. FADH, we know it cannot enter into the first pump, but it can drop its ATP or its hydrogens off at the second pump. So still resulting in an accumulation of hydrogens in the outer membrane. So an electron transport chain literally means a transport chain. So the electrons, this is the electron pathway right here, this pathway that kind of goes through the middle of that membrane. So that's where the electrons will travel, and then the hydrogens will flow across the top of this membrane. And eventually those hydrogens are going to be pumped into this respiratory assembly. So respiratory assembly, or also called ATP synthase complex. And the ATP synthase, that's the enzyme. So the ATP synthase is the enzyme that's going to catalyze the ATP or ADP in phosphate and turn it into ATP. And so we see that down here at the bottom. The videos that I kind of attached up here for y'all to watch, and there's one down here at the bottom. Those were really helpful in showing an actual depiction of the electron transport chain and how the hydrogens are pumped and how the electrons are passed and how we make ATP. But again, go back to the lecture video where I discuss this in a lot of detail. But what you have to know is first the respiratory assembly, and know that the ATP synthase is the enzyme that produces the ATP at the end of the system. Also know that the byproduct down here is going to be water, and I'm not sure if it shows it, it doesn't. But the video shows how oxygen is the final electron acceptor, and that forms water at the end of the cycle, which is the byproduct or the waste product, and helps to regenerize the cycle. And then you also have to know total ATP production at the very end of the electron transport chain from one molecule of glucose or glycogen. And so I have that here. So we know that one NADH equals 2.5 ATP, one FADH equals 1.5 ATP. So we figure out how many did we have to start with, and that gives us kind of our numbers for each one, with our total ATP from the ETC only is 28. And then we add the 4 to 5 ATP that we got from the other steps, giving us 32 ATP if we started with glucose and 33 ATP if we started with glycogen. So again, you just have to know kind of net production of ATP depending on what fuel source we started with, and then also net production from each of the categories there. But yeah, that's going to be all you have to know for the energy systems. So we're kind of done talking about energy systems, and we're now going to kind of move on to the other topics that we discussed in this system or in this chapter. So starting with the Cori cycle, and then we'll talk about the lactate shuttle. So on your review, it basically says no, both of them. They're very similar, but if you just kind of read our definitions here, we know that the Cori cycle takes place in the liver. So it takes place in the liver, and this is where lactate that's produced by the muscle transported to the liver through the bloodstream and is turned into glucose or stored as liver glycogen. So that glucose, we can use again back to the muscle to go through glycolysis. So both of these processes, Cori cycle and lactate shuttle, is just another way to use lactate in the body. So lactate actually shouldn't be seen as a harmful waste product. It's kind of more as another way to transport glucose around the body. And then here is our lactate shuttle. This is similar to the Cori cycle, but again, this is a hypothesis. It's not concrete, it's not a theory, or I guess it kind of is a theory. It's not a law, it's not solid yet. So it's basically just a hypothesis of what else can we do with lactate. So basically it says that lactate is used kind of as a fuel source because we can turn the lactate into glucose, and then the brain can use it, or other organs can use it, or we can send it back to the muscle. So that's kind of what these two I guess concepts talk about, is just, and you look at this picture, how lactate can be shuttled around the body and used for fuel source in different organs and different tissues. So yeah, that's kind of what we have for Cori cycle lactate shuttle. Next, let's move on. The next topic is beta oxidation and protein oxidation. So yes, we're kind of in energy systems here, but I think this is pretty interesting because it talks about how we don't have to just use glucose or carbohydrates for fuel sources. We can use fat and we can use protein in aerobic metabolism, which is unique because we can't do that in anaerobic metabolism. So let's look here, starting with beta oxidation. And I have some good pictures of it that we'll get to. But beta oxidation is just using fatty acids to produce more ATP. But before we talk about beta oxidation, another point here is the role of insulin and the role of lipase. So in order to understand beta oxidation, we have to talk about these other two topics first. So first, insulin. We know insulin is a pancreatic hormone that moves blood sugar, into the cells. So it moves the sugar in the blood, or the glucose in the blood, into the cells. And once we get it in the cells, we can actually use it for energy. But if it's just in the bloodstream, we can't use it. So insulin is a very beneficial hormone that moves the glucose into the cells so we can use it for energy. Next is the role of lipase, also called hormone-sensitive lipase. So this is also an enzyme that breaks triglycerides into glycerol and fatty acids. We know that glycerol cannot be used by the muscle tissue, but it can be used by the liver to make more glucose. So glycerol, we can't really use it, but the fatty acids we can use. So fatty acids is what we're going to talk about here in beta oxidation. So we already said glycerol metabolized, but it's turned into glucose, and it's done in the liver. Fatty acids, though, they can enter into the muscle tissue. So they are broken down into acetyl-CoA, just like glucose was, or just like pyruvate was. But this process here, this is the mitochondrion, and it just shows how fatty acids go through this beta oxidation process. Something you do have to know for the exam is how many ATP are required to start this process, kind of like the investment phase. So how many ATP do we have to invest into this system before we actually see a production of fuel? So it requires 2 ATP. So 2 ATP per fatty acid to start this process. And then we get an NADH and an FADH. So we know that we can use that in the Krebs or in the ETC to produce more ATP. One other thing you do have to know is that ATP or beta oxidation, when we metabolize fats, we produce more energy than when we metabolize carbohydrates, but it takes a longer time. So beta oxidation is a slower process. When we put fatty acids through aerobic metabolism or oxidative phosphorylation, it takes longer, but it does generate more ATP. That's why we use fats for fuel sources at rest, because we don't need energy very fast, but we need it in a large amount. And so that's again why we use fat for fuel sources at rest and carbs for fuel sources during exercise, because it generates a lot of energy very quickly, and it does it kind of fast and quick, a lot of it. But yeah, so that is beta oxidation. Something else you do have to be familiar with, but this one's pretty easy, is this question right here. It's basically on the exam. So can you gain fat mass simply from just eating proteins and carbs? And if you go back to the lecture and watch how I explained this picture right here, the answer is yes. So it's just something you have to know, that protein and carbs, we know if we follow these arrows right here, they can be turned into all these different substrates and stored in the body as something else. So yeah, go back and listen to the lecture if you need more information on that. This picture also does a really great job, and I spent a lot of time in the lecture going over every single arrow and talking about how they're stored and how they're used for energy. But again, yes, we can have increased fat mass just by eating carbs and protein, based on how they can simply just turn into something else and store it in the body. But the next topic is that protein oxidation. So right here, protein oxidation, we see that amino acids can enter into the Krebs cycle. So they can enter straight into it and still produce ATP. So let's talk about the 2 ways that we can metabolize protein. And you do have to know this for your exam: transamination versus deamination. So we talked about how the first protein, amino acid. So we have an amino group and we have an acid group. So right here we read transamination, and this is when the amino group is transferred to a keto acid. And that's how we can use those keto acids to produce ATP. And then the other way is deamination, which is shown in this picture right here. So again, with our amino acid, we remove our amino group, and then we transform that amino group into ammonia. And then we add carbon dioxide because we have some sort of electrical imbalance when we have ammonia. But if we add CO2 to it, we have water as a byproduct, and that produces urea, which is just kind of transferred into the kidneys. So I know I'm not going to spend a lot of time going over those two topics here, but they're talked a lot about in the lecture. So yeah, you just have to know the difference between transamination and deamination. So continuing kind of with factors that influence when protein is used in the body for fuel, right here, this slide. So we discussed that it is not beneficial to use amino acids for fuel. This is not something we actually want the body to do. But when does it occur? We said it's really small. Extreme dieting. So when we don't have enough fuel for our body is going to have to pull from something, and it may pull not from our fat tissue, which would be ideal, but from our muscle tissue. And so that's one thing we don't want to happen. Another is high protein diets. This goes back to the availability factor. So if that's what's available, if that's what's abundant, then our body is going to pull from what is easily available or easily accessible. And then long-term endurance activity. So this activates that enzyme we said called proteases, and this enzyme breaks down and metabolizes our amino acids so we can use them for energy. Again, this is something we don't want to happen as well. So long-term endurance activity, that's why it's important to consume fuel, to consume a glucose-like source so the body can use that for energy rather than turning to our muscle for energy, again, which is not what we want to happen. Okay, so kind of continuing with getting into like the endurance activity part and exercise and when carbs and fats are used. This right here, I think it's on the quiz that's open today, and it's going to also be on your exam. It's just understanding at rest versus maximal intensity, how much of our ATP comes from our fat sources and how much comes from our carbohydrate sources. So yeah, again, I talked about this a lot in the lecture, so go back and look at this, but those numbers are very important. So if I were you, I would know those numbers right there for rest versus maximal intensity. All right, so continuing, this is one of my favorite topics, is talking about during exercise. So what happens when we run out of glucose, when we run out of glycogen? That term, hitting the wall, you will also see on the exam. So what does it mean to hit the wall? Glycogen depletion. So it has nothing to do with oxygen. It has nothing to do with ATP. It has to do with glycogen. It's when our muscles run out of that stored glycogen. So yeah, we talked about ways that we can prevent hitting the wall, and that's consuming carb-like sources during exercise, especially the long duration. So greater than 60, probably more so around 90 minutes is when the body would need that extra fuel. So we don't tap into all of our glycogen stores, and which would decrease performance. So yeah, we'll talk more about glycogen in a second, but you have, I think, about 2 questions kind of from this slide right here. So this is where we have to think big picture. We have to think like situational kind of questions. That's what you're going to see. So stuff like, okay, if I consume this, or if I don't consume carbohydrates and I'm exercising for this amount of time, what happens to insulin and what happens to lipase? So we have to understand the role of insulin, lipase, and carbohydrates and how that all works together. So one example here is, let's say that I consume a lot of carbs before exercise. So I am loaded up on carbohydrates, meaning my body has a lot of glucose and glycogen to use for energy. So what happens to insulin? Insulin is going to be high because when we ingest a lot of carbs, we have to have insulin to move it into the bloodstream. Lipase though, lipase is going to be low. We see here insulin inhibits lipase. So lipase breaks down fat. But if I have a ton of carbs to use, my body won't necessarily turn to fat stores until either the intensity decreases or I run out of those carbs. So again, the questions you'll see about this slide here, they're situational. So I give you something that happens and you have to explain what's the result of that. Next is talking about muscle and glycogen stores. So I talked in the lecture how many grams of carbs we have, or sorry, how many grams of muscle glycogen we have stored and how many grams of liver glycogen we have stored. So we see numbers right here. I think I mentioned in the lecture, depending on body size, you look down here, this is a 143-pound person with 12% body fat. So that's where these numbers come from. But generally, it's going to be about 400 to 500 grams of muscle glycogen stored in these little granules. And then liver glycogen, it's about 100 to 120, or you see right here, 110. So it depends on body size. So let's talk about the factors that influence glycogen storage. And number one is body size. So larger bodies store more glycogen. Number two is diet. So are you actually consuming carbs? Is that part of your diet, or are you on a low carb diet? And then another is exercise training. We talked about some adaptations to our glycogen stores where we can increase them, or they can increase depending on how we train, if we train in a fasted state or a fed state. All of those factors influence how much energy we can store in our muscles. So yeah, you'll see some questions like that on the exam. Next is lactate threshold versus OBLA. So you answered this on your assignment. We talked about why is lactate threshold, having a higher lactate threshold, why is that important for an aerobic athlete? First, we have to know the definition of lactate threshold. And that definition is when blood lactate increases above baseline. So we say here baseline is very low. It's like 0.05 or something around there. And when we start exercising, we know lactate's going to build up in the bloodstream because we're using fast glycolysis. That lactate's going to build up. So when that lactate builds up and when it increases above baseline, that's where lactate threshold is. And then lactate starts increasing rapidly. So that's where OBLA comes in. Remember, OBLA is a set number. It's at 4 mmol per liter. And we see right here, there's 4 mmol. So that's where OBLA occurs. OBLA is the same for everybody. It's lactate threshold that's different. So how does training influence lactate threshold? It pushes it to the right, meaning that we can basically clear lactate away. So trained athletes have increased lactate clearance, meaning they can clear lactate away, send it to the Cori cycle to make more glucose, decreasing the blood lactate levels, allowing us to exercise at higher intensities for longer periods of time before we hit fatigue. Remember though, lactate is not the cause of fatigue, but it is associated with it. So they're associated, they kind of occur together, but it's not the direct cause. So yeah, that's what you have to know about lactate threshold and OBLA. But since we're on aerobic adaptations, let's move to all of the adaptations that we see with chronic aerobic training. I was actually really disappointed at some of the assignments that it asked about aerobic adaptations and why we would see certain adaptations with aerobic exercise and not with anaerobic. And I really didn't get a lot of good answers. I mean, for some people, yes, I got great answers. But this slide is kind of where you wanted to turn to answer that. So one of the adaptations we just discussed was the increase in lactate threshold. And lactate clearance. So clearing lactate away, improving our threshold to where lactate stays low while we can exercise very high. Next is enzyme activity. So aerobic enzymes are going to become more efficient. We wouldn't see that with anaerobic training. Next is substrate availability and substrate use. So this is just improved ability to use certain substrates for fuel rather than others. So the example here was a greater reliance on lipids, which are fats. That allows us to spare glycogen, where we can save up our glycogen stores. We don't have to use them, and we can rely on our fat stores more. So trained athletes, their body has adapted to that chronic exercise to where they can pull from their fat stores and save their glycogen. The benefit, it helps to delay fatigue. And then we talked about other physiological changes like improvements in mitochondria, improvements in capillary density, blood flow, oxygen delivery, kind of all of that. So yeah, those are our aerobic adaptations. So next, let's get into oxygen deficit steady state in EPOC. You probably already know your chapter 3 quiz review. That's today. I know some people have already taken it. Closes at midnight. You've already probably seen this. So yeah, be sure to take that quiz today. So we're in the last part, meaning we're kind of almost done with this review, and then y'all can ask questions. We want to talk about the process of how our body kind of uses oxygen from the start to the end of exercise. So first starting with oxygen deficit. So oxygen deficit, we know, is this pink box right here. Hold on. So this pink box right here, this is basically where the body, there's this delay in oxygen uptake because our oxygen or our ATP is being produced anaerobically rather than aerobically. So our body is not very efficient at using oxygen right when we start exercising. So there's this delay. Basically, the oxygen that we need, or the ATP that we need, is not being met aerobically. So what is this influenced by? It's influenced by the type of exercise you do, which you can see here. There's a very small delay when we do easy to moderate exercise, but the harder and more severe our exercise becomes, the greater deficit there's going to be. But I want you to think about this practically. Think about the last time you did aerobic exercise. So the last time you jumped on a treadmill or a bike or started doing any type of aerobics. You probably noticed that your breathing was kind of faltered at the beginning, where it wasn't very efficient. It was either really fast or really deep, or just kind of uncoordinated breathing patterns. But once we kind of hit this level out point, we realize that we're at steady state, and that's what shows right here, where all of our ATP is being met aerobically. So this is when our VO2 plateaus off. So you see in this picture, it levels off. So steady state is also an exercise intensity that we can maintain without having a drastic change in our oxygen consumption. So it's where oxygen consumption is steady. This is also influenced, like I said, by training status. This picture here shows that the more trained you are, the faster you can reach steady state. Untrained you are, it takes a little bit longer to get there. And then next is talking about EPOC. So this is when we end exercise. So it's excess oxygen consumption or excess post-exercise oxygen consumption. So it's oxygen that we consume above baseline, even though we're already done with exercise. What is this more influenced by, intensity or duration? You probably know the answer, it's intensity. The harder we're exercising, the greater the EPOC will be at the very end. Duration has some impact on it, but if duration is combined with high intensity. So if you go for like an easy 5-mile jog at a very low pace, you probably won't be or have a lot of EPOC at the very end. Versus if you did maximal sprints back to back to back, your EPOC is going to be very high. So EPOC, we know it's where we're consuming excess oxygen to decrease our heart rate, our blood pressure, decrease our temperature, clear away acidity. So basically returning to homeostasis. So it's getting back to our rested level. And then this picture here just kind of puts it all together. Let's move on to active versus passive, something else you have to be familiar with. So passive recovery is when you do no cool down basically at the end of your workout. So you do a workout and then you do no cool down. Active recovery is basically light to moderate exercise after you're done, which is our cool down. Active recovery is more efficient. It's more efficient at decreasing blood lactate levels, clearing away that acidity, and getting you back to homeostasis faster. So yeah, knowing the difference between those two. Next is getting into RER. So with RER, we are respiratory exchange ratio. So this is percentage of carbs and fats used for fuel during exercise, and it just puts it into that ratio. We know that protein is not used in the RER calculation because we don't ever really want to use protein, and even if we are using it, the percentage is very low. So this chart here puts the RER into perspective because you already know these percentages here. We know that at rest, only about 33% of our energy comes from carbs and 66% of our energy comes from fats. So that ratio is 0.8. So you do have to know that. Next, maximal intensity. You already know that if you're exercising as hard as you can, maximal intensity, you're using about 100% carbohydrates for fuel. That is 1. The ratio is 1 here. But yeah, that's all you have to know for the ratios, just knowing what they equal rest versus max intensity and those percentages. All right, next is indirect versus direct calorimetry. I actually don't think this is on your review, but it's on your exam. So this is added. There's a question about indirect versus direct. So we know right here, this is indirect. So this is an estimate. So when you're hooked up to a gas mask and you're running on a treadmill and we're getting those RER values, that's an estimate of your energy production, and it can tell you how many calories you burned during that exercise, basically based on the CO2 and the oxygen that's being passed. And then over here is direct. So this is our direct calorimetry. This is inside a chamber surrounded by a water jacket where we're exercising. This one here is very, I guess it's, what's the word? It's hard to do because not many people have these chambers. It's not as realistic to be able to assess energy production here. What we do in our labs in this department is a lot of indirect measures of calorimetry. So just knowing the difference between those two. Kind of last slide here is understanding BMR. Kind of RMR, BMR, same thing, very similar. And this number right here. So percentage of calories that we can estimate from an RMR or a BMR value. So if we get a number, let's say that our BMR is, we'll say 1500. So 1500 calories that we burn at rest, 60 to 75% of that basically gives the estimate of kind of the total calories in the day. What are the other two factors that influence that make up the 100%? One of them was exercise. So exercise will increase our caloric burn. And then food, the thermic effect of food, because we actually use energy to digest food. And so those are the other two values that make up the total percentage there. But yeah, so that's kind of all I'm going to talk about with the lecture, and then I'll pull up that review again. Hold on, let's share screen to this one here. Let me try again. Let's see. While I'm getting this up, if anyone wants to kind of start asking questions, okay, yeah, if anyone wants to ask any questions about this review or about your exam on Monday, I'll kind of pause and I'll just kind of wait patiently and see if anyone wants to talk about anything. If you notice down here, there are a few things added that weren't on yours, which were these two right here at the bottom, and then I think one more in there. But yeah, I will just wait and y'all can ask questions and we can talk about anything you would like to. one thing to just kind of keep in mind with the exam is some questions are application questions, like this one here at the bottom. So it's just kind of understanding if I deplete a certain fuel source, that means I was doing what type of exercise. Or if I'm doing this type of exercise, and if I'm in a fed state, which just means I am fed, I ate a lot of food or X amount of energy before I started my exercise, what fuel source will be depleted? Or if I'm in a fasted state, haven't consumed anything, what fuel source will most likely be depleted during a certain type of exercise? But yeah, there's 50 questions. And you know that it also involves chapter 2, 50 questions, multiple choice, true, false. There are a handful, maybe like 5 out of the 50, which are fill in the blank questions. And just remember, if you think you got it right and it counted it wrong, I will always go back in and correct that after everyone's taken it. Same with the quizzes today. There's some fill in the blank on the quiz. So if you notice you got it wrong, but you should have gotten it right, don't worry, I will go in and correct it. But yeah, I'll wait just a little bit longer for anyone you can type a question in the chat box, or you can unmute your microphone and ask a question. But yeah, that's what I'm here for. So again, I will wait just a second. Okay, well, if there's no questions, that's completely fine. I understand. You can always email me a question that you have, or you can ask to Zoom with me individually, and we can, again, talk about anything you want to talk about. But yeah, that's all I have for you then. So good luck on your quiz today, and good luck on your exam on Monday. And then we'll get started kind of with the rest of next week. But yeah, thank you so much for being here. I really appreciate it. It means a lot that you took this time out of your day to come listen to me talk. So yeah, thank you, and y'all have a great weekend.